All right, at my watch, it's 6 p.m. Nashville time, so we will get started. As I mentioned, we will be recording this presentation so that we can put it on our website and make it available to the entire School of Engineering community, including on Wednesday through Conduit. So I'd like to thank the development team, Jen Schad, who is right there and has organized this, and Teresa Rogers, who is a little bit under the weather and will not be attending this meeting, as well as the School of Engineering staff member and faculty who have made this meeting possible. Since we are recording this, I'm Philippe Fauchet. I'm the Dean of the School of Engineering. Today is Friday, April 24th at 6 p.m. in Nashville time. I'd like to welcome all our Board of Visitors members, as well as the School of Engineering community members to this 30-minute or so presentation. Presentation on the School of Engineering response to the COVID-19 pandemics. As of today, Vanderbilt has been on remote work for about 6 weeks, which means the campus is deserted. Yet research related to COVID-19 has been allowed to continue on a limited basis to ensure that all the safety measures such as social distancing, etc., are implemented. Now, as you know, engineers in general are very active in a wide range of projects, and related to COVID-19, we are active in a wide range of projects ranging from diagnostics, research on diagnostic tool, research on biologically inspired response, on device development, on building aids to patients affected by COVID-19, and data science and computer science approaches to prevent or monitor, at the very least, future pandemics. When I asked the faculty and the students how many projects were going on right now on a limited basis, there were about 15 to 20 projects, ranging from the complete gamut of research we do. And so what I did is I selected about 6 of them for presentation today to give you a range of what we do. And I want to thank all the researchers will be highlighted today, all the people who work with them, as well as all the other people who cannot be highlighted today. The 6 presentations, we will start by 2 presentations, 2 very different presentations coming from the Vanderbilt Biophotonics Center. Associate Dean Senior Associate Dean Duco Jensen will introduce the 2 speakers there. Then we'll move to BME Professor Rick Hazelton's group. Then we'll move to 2 different projects originating from the Vanderbilt Institute for Surgery and Engineering, VICE, one from a faculty member, Bob Webster, and one from a graduate student, Kathy Riojas. And finally, we'll have a presentation from Professor Janos Stipanovic, who is the director of our Institute for Software Integrated Systems. I'd like to thank all of you. And without further ado, I will ask Senior Associate Dean Duco Jensen to introduce the 2 projects from the Vanderbilt Biophotonics Center.
Great. Thank you, Philippe, and it's good to see everyone. So the Biophotonics Center has a lot of resources that are normally being used for our photonics research, and we very quickly pivoted those to make them available for 2 projects in particular that we're going to talk about now. So the first project is a very heavy computational project that's essentially using the resources from, to a large extent, our TIPS program in high resolution imaging, lots of data, lots of computational power that we have. So that's going to be presented by Brian Millis, who's a research assistant professor in biomedical engineering, also associated with the Data Sciences Institute and the Cell and Developmental Biology department. And then the second project is going to be discussed by Anita Mahadevan Jensen, who's the Orrin H. Ingram Professor of Biomedical Engineering, and she's the director of the Vanderbilt Biophotonics Center as well. So without, you don't want to hear from me, so I'll just turn it over to Brian Millis.
So thanks for having me give a talk today. I'm historically very long-winded, so I did everyone a favor by recording just a few slides to make sure that I stayed with under the 5 minutes and was respectful of your time. So let me see if I can share my screen. You may have to bump your audio just a little bit, but let me know if you have any problems when I share my screen, and we'll get started here. Can everybody see my screen?
Yes.
Okay. First, I just wanted to thank Dean Fauché for giving us some time to talk to you about a couple of projects within the Vanderbilt Biophotonics Center with respect to the current COVID-19 pandemic. So you're all likely familiar with the CDC's image on the left, which displays a rendering of the surface of the COVID-19 virus. If you look at these prominent red protrusions in this rendering, they kind of look like origami. These are actually proteins, and the folding of these proteins is incredibly important for their function. We can, as scientists, predict the three-dimensional structure of these proteins, and therefore that gives us a lot of clues in terms of how to go about designing and modeling effective vaccine targets or drug targets, for example. However, the biggest problem with prediction of those of that structure and conformation is the actual calculation process itself. So these are extraordinarily complex calculations to run. Such that if you were to run them on your home computer, for example, it could take hundreds of years to get a result. So enter the world of supercomputers. You're actually looking at the world's fastest on-site supercomputer, which is housed at Oak Ridge National Laboratory, not too far from Vanderbilt. It can perform on the order of 200 quadrillion operations per second. So then it might sound a little ridiculous if I were to suggest to you that that massive computer you just saw couldn't even come close to competing with my little laptop sitting here on my dining room table. Now, of course, I'd only be telling you a partial truth here, because that little laptop is actually part of a distributed supercomputing project whereby anyone can donate time on their computers to run simulations of protein dynamics. So the idea here is that while you're sleeping or while your computer is idle, you can download an app and have it run in the background simulations for this project and contribute to a greater cause as a part of a worldwide supercomputing network. And what results here is some staggering statistics. It's by far the most powerful supercomputer on the planet. Recent estimates calculate that the Folding at Home project has more compute power than the top 100 supercomputers in the world combined. Interestingly, this has grown exponentially as a function of the COVID-19 crisis. So the Folding at Home project has been around for about 20 years, working on a number of diseases related to protein folding and or misfolding. And up until about a month or two ago, they only had about 30,000 users and still doing phenomenal work with that number of users. As of March 30th, they had over 1 million users contributing to these calculations. To give you an idea of some of the non-COVID related diseases or conditions that they study, I'm showing there on the left, which you can read. And you're starting to see Folding at Home show up in mainstream. And if you guys haven't heard about it already, I'm sure you will be hearing about it because it's making quite a splash. There is a fun side to this as well, a little bit of a competitive side. So with the 1 million users, there are people actually for years that have been competing as teams to try to accumulate points. So for any of these calculations, points are assigned based on the complexity and the length of the calculation. And so people gather together and have a bit of fun with this and compete between institutions for how many work units, they call them, or points that you can accumulate. So we've only been doing this within the Vanderbilt Biophotonics Center for about 3 weeks since March 30th, when we officially launched our team. But we're currently ranked 143 out of 12,000 teams if you look at the average points accumulated per week. We have blown past quite a number of recognizable institutions, so we're having a bit of fun with this with our colleagues. But obviously there are some big players in this, as you can see on the right-hand side of your screen. So a little bit of good fun that we have and competitive nature for a good cause has been quite a reprieve in this time of otherwise really stressful stuff as well. So we obviously have a lot of computational power for reasons that are very different from simulations of protein dynamics. So I do want to take time to recognize the people here for putting up with me. It's not a small feat to repurpose your instruments, which are otherwise sitting idle while we're all sitting at home, and can't be on them. So these people were either directly involved or supportive of us utilizing our resources for this great project. And I just want to give them a thank you.
Thank you, Philippe. That's the conclusion there.
Thank you.
All right, so now we'll turn it over to Anita Mahadevan-Jansen.
All right, so everybody hopefully can see that screen. There we go. So I'm Anita Mahadevan-Jansen. I'm the director of the Vanderbilt Biophotonics Center. And I wanted to show you a completely different side. As Duco mentioned, a lot of what you're hearing is a result of investments from the Trans-Institutional Program at Vanderbilt to start what we call BioMid, or it's about innovating biomedical microscopy technologies. And for bringing
Anita, this is Brian. Your audio seems to be a bit muffled. Is
this better?
Yeah, that's perfect.
Okay. And so BioMid essentially brings a cutting edge microscopy to Vanderbilt that's not available commercially. And as a result of that, we've developed a lot of technologies, 3D printers, of course, which we used for prototyping and use components to mock up what our microscope might look like and test that it works before we build the actual microscope that you see here and the kinds of images that we can get to give you a fine level of detail on biological processes. Of course, that means you need computational power to be able to build those images, which is just how we're using now, given that this microscope is currently shut down and not operational for users at Vanderbilt. We're using the core pieces of it to make that work. So the other piece was the 3D printing that I mentioned. Well, clearly PPEs and masks especially have seen a lot of buzz in the recent few days and few weeks. And so one of the things is, of course, personal, not just by the frontline workers, but in every person that CDC just declared should be considering wearing masks. So we started thinking about how we could use our 3D printers to help here in this domain. And instead of reinventing the wheel, we discovered this Montana that was designed at the Billings Clinic in Montana that actually provided the template file needed to do the 3D printing of these masks. We just had to think a little creatively about how we wanted that filtering window to be equipped. We really want to cut up, existing masks to fit that, and we can do 5 or so more of those, cut them up and put it in that window. Some creative thinking about the elastic band needed to actually put it in place. So given that, here you see where you can see one mask being 3D printed using our just standard benchtop printer. This is not huge machines, these are just benchtop, about a foot by foot big, taking about 7.5 hours to print masks. But we decided to be creative, sort of like a jigsaw puzzle. So how many can we really print in a single run? We can do roughly about 5 of different sizes along with the filters. Remember though, we are a prototyping group, not a production group. So we're really trying to see what we can do to help the community. Of course, then you print those and then you can assemble them by being a little creative and sort of putting this together. The one thing we discovered by scaling the design, we could print them in different sizes. So here you see Brian's daughter wearing one with, pink being a little girl's favorite color there. So we could use our pink material, PLA material to do that and be also cool and funky. So a variety of different sizes that we can indeed do. So we've been printing for several days now, weeks, I guess now, and then those of our operational folks who go and clear that out and collect them for us. We could print them in all sizes, from adults to kids. We were informed by the VUMC that the frontline workers are certainly well equipped right now, and indeed these masks have not been approved, the long testing needed for approval to actually use it in the front line. But there are plenty of other organizations, and including our own selves, that need these masks. So we have so far distributed these masks to a local nonprofit called Renewal House, who specialize in treatment of women recovering, mothers recovering from substance abuse. Such a program obviously does not push the pause button and stay home. It continues on, and their counselors and staff clearly need protection, and we've been providing them for that. We will continue to produce these with the intention of distributing them amongst those with needs. And certainly as we start to talk about starting back up our research facilities, we would be able to provide them for our staff and faculty. With that, I want to thank Brian for pushing this, for making this happen. Certainly you can see the folks at Renewal House wearing them here as well, and thank you very much.
All right, thank you. We'll take questions at the end, if that's okay. The next presenter is Professor Rick Hazelton from Biomedical Engineering. Rick, it's yours. Rick, you are muted. We can't hear you.
Thank you, Dean Frichet. Thank you for the opportunity to talk about our research, always one of my favorite topics. My colleagues and I have been working on diagnostics for infectious diseases such as malaria, TB, HIV, and influenza for about the last 15 years. Most of our efforts have focused on developing simple designs for global health applications. Of course, many folks have helped develop and test these designs, including, in particular, BME professors Mindy Liu Wong and Nick Adams. And it's a pleasure to introduce Professor Nick Adams, one of the major contributors to these efforts, to share with you how we became involved in the COVID-19 effort. Nick?
Hi, good evening. Like Rick said, I'm Nick Adams, Research Assistant Professor in Biomedical Engineering. Thank you for the opportunity to talk about some of our work. I have some links to some of the work that we've done in the past that I'm going to paste into the chat here so that you could check it out if you'd like. I can display this behind me, I think. I don't know if you'll be able to read it with my head in the way, but maybe that'll be helpful. So I'm excited to talk about some of the stuff we're doing with COVID-19. I'll give you some background about what got us into COVID-19 research. So during the development of an influenza diagnostic device that we've been working on and is currently being tested at the Tennessee Department of Health, we made some observations that were key in the COVID-19 response, which is that the RNA extraction step, which is known to be a critical step in the diagnostic process, might not be necessary. And this is important because the RNA extraction step is a bottleneck in the diagnostic process of most all molecular testing, including that of COVID-19. So if you're not familiar with what molecular testing is, it's the most common method for testing COVID-19 among many other infectious diseases. And it works by identifying the presence of specific DNA sequences or RNA sequences in the case of most viruses in a patient sample. So the first step of doing this type of test is after a sample is collected, and I happen to have had a sample of my own collected, so I have a photo of that here. But once that sample is collected and the swab comes out of your nose and into that liquid preservative that they put it in, it comes with it not just any potential virus, but also salts and other debris, cell junk, that's inside of that solution. And so it's required that you purify the RNA from that sample prior to the next step in the process, which is to detect the RNA, the purified RNA, which that step is called polymerase chain reaction, or PCR. And you do that RNA extraction to make sure that PCR is not inhibited by the contaminants. But I described this RNA extraction step as a bottleneck because it's very expensive, time consuming, and as it turns out right now, it's very hard to get a hold of the kits to do RNA extraction. There's a shortage right now all over the US and the world, and it's holding up testing in a lot of labs across the US and in other parts. So this observation that we made while working on our influenza test, which is that we could get rid of the RNA extraction step, is very important. And we were able to do this because we've put together a special cocktail of ingredients for doing the PCR step, the step that's used to detect the RNA, that's tolerant of those background salts and nasal debris and other contaminants that come from that raw sample. So we took this idea of testing this streamlined diagnostic process where the extraction step would be omitted to the director of the Vanderbilt Medical Center Diagnostic Lab. And he was thrilled at the idea of getting rid of this RNA extraction step. Not only was he having a hard time getting a hold of the numbers of kits that he needed, but he felt like it could increase his testing capacity to meet the demands that he knew were coming. And so he was eager to set Mindy Lilawang and I up with the appropriate certifications to come test our method using some residual patient samples that he had stored in his lab of COVID-19 samples. So Mindy and I worked with him over the last few weeks in his lab, testing more than 200 positive and negative COVID-19 samples. Here's a little picture of me over there. And what we found is that this approach actually works really well for coronavirus. And it was successful, and we feel like this can have a major impact on helping keep up with the demand of testing in labs across the United States and across the rest of the world. We're now writing up a publication of these efforts that we've done with COVID-19 over the last few weeks that we're submitting soon so that others can benefit from these results. And of course, we've also submitted grant applications to scale up this research to the CDC, and we have several more applications lined up to submit to the National Institutes of Health in the coming weeks. So these are exciting times to be working on the development of viral diagnostics, and we're happy to be a part of it.
Thank you, Rick, and thank you, Nick. Appreciate that. If we can go back to the normal mode. The next two presentations are one way or another related to the Vanderbilt Institute for Surgery and Engineering. And the first speaker, I think, will be Professor Bob Webster, who is the Richard Schrader Professor of Mechanical Engineering.
All right. Thank you very much. Can everybody see my screen now?
Yeah.
All right, great. So I'm going to tell you guys about how we built a ventilator from scratch, literally in our garages, a little bit on campus, but a lot in our garages. Looks like this. It's an open source ventilator that basically anyone could build. If you have a wood shop in your garage and you have soldering skills, you could put one of these together. So let me just take you through the story of how this came about. So the question I want to answer today is, how did we go from napkin sketch, which is what you see on the left, to this fully functional device in such a short time? So napkin sketch to an animal trial in 2 weeks, to a successful animal trial in 3 weeks, to an FDA submission in 1 month, and to 100 units made in just over 1 month. So I'm going to take you through how this happened. So it started out March 21st. This is Duke Harrell. He's a doctor at Vanderbilt Medical Center, who's one of the leaders in VICE on the medical center side. So he called me, and as you guys all probably went through the same progression I did in your minds about what is COVID and how bad is it going to be. And, you know, initially it's just over in China. We're not too worried about it. Then you're a little more worried. Then, you know, Italy's getting bad. Well, at some point for all of us, we realized this was really going to be bad. And for me, it was when Duke called me, and that was March 21st. And he said, hey, we could easily run out of ventilators in Nashville. And I'm like, well, what's a ventilator? And he's like, well, people who have COVID who need these die if they don't have them. And that's the problem in Italy. And that's why so many people are dying. This might happen right here in Nashville. And ask the question, is there anything we can do? I mean, we're, you know, engineers and doctors who've worked together a long time. What could we do? Could we build one of these? Is it even possible? So he asked me that question. And that got me really moving on this. And so I'm going to start off March 21st, and you'll see up at the left, upper left side of the screen, the date. And so just track with us through this amazing whirlwind of a project. So the first thing I did is I called Eric Barth, because he's a fellow mechanical engineering faculty member. So I knew he had been exploring some of this stuff the week before. So I called him and I'm like, hey, you know, did anything pan out? Do you have any specific ideas? Like, what can we do? He's like, well, Kevin Galloway does. And Kevin Galloway, as some of you know, is the director of making at the Wondery. And so Eric told me that Kevin had a napkin sketch. So I called Kevin, and Kevin sent me this. This was his napkin sketch. So for Kevin at the time, it was just like, well, you know, I'm just toying around with this in case maybe we might need to do it. So he wasn't really planning on building this at the time. But I said to him, what would it take? Can we build one? How quick can we do it? And we started working on it. We quickly got some pulmonologists from Vanderbilt Medical Center, Fabian Maldonado and Matt Bacchetta, involved with the team. So we had really good clinical advice, and then some super talented graduate students, one of whom is Katie, who you'll hear about next. She's been involved in both projects, but especially Josh Gafford did a lot of work on this. And so these folks worked literally night and day on this project to get it where it's gone. So March 22nd, notice the date again in the upper left. March 22nd, this is what it looked like. So this is the day after I first called Kevin. This is what he hacked together in his garage that night. I went over there and we hooked up, this is a windshield wiper motor. We hooked it up to some, you know, nylon straps, and we said, hey, we could put the bag up here and pull on it. The very next day, we got one of the bags from the medical center and we started pumping it. So this behind here is the windshield wiper motor. It's just spinning continually, and you can see how it's pumping air. So the patient is where the balloon would be. That blue balloon is the patient's mouth. So that's March 23rd. So, you know, we're 2 days in right now, and we've got a device that kind of does something. Then the same day, Kevin was like, I think I can do this better. Why don't we squeeze the bag with between two, these are pool noodles, between two surfaces, because that will be more reliable, because we couldn't figure out how to set the amount of compression with the straps in an easy way, because doctors need a fixed amount of volume to go into the patient each pump, and they need to be able to set that. So March 23rd, we had this design. March 24th, we had another design, which we'd then run for 20 hours. This is a little bit smaller. This uses a motor from Nissan. So we actually got a donation of 100 motors from the local Nissan plant and built this nicer, smaller prototype, ran it for 20 hours. So we're now 21st to 24th, we're 3 days in. So this project is going lightning fast. 25th, we're over at Vanderbilt. We're testing it out. This is a mannequin they use for training doctors how to take care of patients. And we're testing it with pulmonologists and emergency medicine doctors. And also you can see in the bucket back there, that's an ex vivo pig lung. So we're testing it also in animal tissues. So that's 25th. So we're 4 days into this project, we're already testing. 29th, this is all the electronics. So we've done the control, we put the sensors on, we wired things up, had the electronics done. April 3rd, we go to do our first animal experiment. So we're now 2 weeks in. We do the animal experiment and it doesn't work. And the reason it doesn't work, we found out through doing this experiment, is we have a long tube here between the animals under this blue blanket, and we have a long tube between the bag and the animal, and the valves are back here. And so there's all this compressible dead space in the tube, and that's making it so the animal's not breathing well on our ventilator. So 1 week after that, we do a second animal experiment, and you can see now we've moved the valves right down here near the pig's mouth, and that made everything work great. And so you can see everyone's body language here. We ran this animal for 4.5 hours on this ventilator. So obviously it was not, super exciting like the whole time, which was great because it meant the animal was doing well the whole time and blood was being oxygenated. All of the vitals were normal. And we asked the doctors after this experiment, would you use this on a patient if you ran out of ventilators? And they said, absolutely, we'd use it today if we ran out of ventilators at Vanderbilt. So that made us feel pretty good. So where are we today? Well, we've done, it's basically the same design. We've put a housing around it so you can't pinch your fingers in it. We've made the electronics box look a little bit cooler and nicer. We've got our sensor now integrated in a better spot. So it's been minor changes since then, but it was essentially all done to the point that it could save a life at the 3-week time point. And so as of actually today now, we have 100 units constructed. We've put all the designs open source online. So if you go to vandivents.com, you'll find all the plans. You could literally go in your garage tonight if you have a wood shop, and you can make one of these yourself. And we've applied yesterday for FDA clearance on this device. And man, it's been amazing since we put up the webpage and got some press on it. We've done interviews for all the local TV stations and the newspaper. We've had inquiries from all over: Afghanistan, Bangladesh, Australia, Philippines, all over the world, Kenya. And so we actually think, thankfully, it doesn't look like we're going to run out in Nashville. But what this could really be good for is being built locally wherever it's needed all over the world. And we purposely designed it to use parts that can easily be sourced wherever you are. And so here's a picture from today. This is in the Wondery. Here's, it looks like about 50 units of the 100 that we've built, all set up and ready to go. So if they're needed at Vanderbilt, we can run them over. If they're not needed at Vanderbilt, we'll start sending them to people who do need them for evaluation purposes, and see where we go from there. So that's my presentation.
Thank you, Bob. Kathy, I think you're going to be next.
All right, thanks. That's very cool. All right. Can everybody see that?
Yep.
All right, great. So my name's Katie Riojas. I'm in Dr. Webster's lab at Vanderbilt. I'm a 4th-year PhD student in mechanical engineering. And our lab, our lab focuses on developing medical devices to basically make people's lives better, whether that be patients, surgeons, doctors, etc. So as it looked like things were starting to get pretty desperate, like this picture here, I realized this is why we do what we do. When the doctors at Vanderbilt were expressing concerns, the doctors that we work with all the time, I realized we should try and jump in and help, because this is what we trained for. So one resident in particular reached out to me and started emailing different ideas, and she wanted to know if there was some way that the School of Engineering could help combat the predicted shortages at Vanderbilt or in Nashville in general. And so I spent about like a week looking at kind of what everyone's doing all across the world, doing a little bit of lit review to see how Vanderbilt could fit into that puzzle. And I found the University of Wisconsin's face shield design here. And upon looking a little closer at the design, I was immediately excited. And that's when things really started to take off. So the neat part about this design is that anyone can build it in their own home while maintaining social distancing. This is me in my home building it. And it doesn't require any specialized equipment, can be made with just local materials, and can be made in just under a minute per shield. And so I emailed this link to the doctors on Friday. On Saturday, I went to Joann's and got enough supplies to make some prototypes. And by Sunday, the School of Engineering, the Med Center, and various labs were all involved, and they were providing supplies and incredible support. And I was very amazed at just the big hearts of everyone that I talked to and the fast action. We distributed kits to 2 different doctors and then various grad students to be able to make these kits, everyone in their separate homes while maintaining social distancing. And so with the help of everybody shown here and others, we made a little over 1,000 in under a week. So pretty exciting as far as, like Bob was saying, timeline goes. And on the left here is shown a completed batch of face shields. So there's, I think, around 200 there. And so that was just made in our house here. And then on the right are 2 of the clinicians at Vanderbilt using them in the home.
Okay. Kelly,
And so current status is that we think that the commercial oil will come, just they don't. Sorry?
okay. I'm sorry, were you done?
Yes, yeah, sorry. Did you
Okay,
say
that's
something? I
fine.
missed it.
Thank you, thank you. And then the last presentation is going to be something different, and the presenter would be Professor Janos Stepanovic.
Janos, you're muted. We can't hear you.
Yeah. Yes. Finally, I'm unmuted. Can you guys see it now?
Now we can see you.
Okay, then. Some more haggling needed. Yes. Now you can see it, right?
Yes.
Okay. So let me just talk about this project, which is the Microsoft Premonition project. First of all, let me introduce you Ethan Jackson. Ethan Jackson is a senior director and principal researcher at Microsoft Healthcare. He is one of our graduate students. He was one of our graduate students graduating in 2007 in our CS program. After that, he went to Microsoft. We continued working together. And one of the adventures that created quite a stir is this so-called Premonition project. So as you know, the state of the biome impacts every society, economy, and the government. Disease epidemics are the most catastrophic events in recorded history. Therefore, it's quite important to understand and possibly predict where diseases might emerge. The most frequent hosts of diseases are animals. So it's quite obvious that effective method for preventing, predicting epidemics would be to develop a method for surveillance of animals in the environment. Now, the question is how to build such a surveillance system, possibly in global scale. Ethan came up with a quite interesting idea. And this idea was to use mosquitoes as devices to gather data in scale. The nice thing is that that mosquitoes sample pathogens in animal populations. They like to do it, and they have a pleasure to do this. Well, this idea landed for Ethan in 2014, the first MSR, Microsoft Research Expeditions Award, and the project started up. In a year, we joined effort. We organized together an academic team, which was parallel with the Microsoft research team. We led the academic team, and the other partners were Pittsburgh, University of Pittsburgh, Johns Hopkins, UC Riversides, the Harris County Public Health Department in the Houston area, IARPA, and Microsoft. This is the mandatory quad chart for all of these DARPA-like projects. So the only thing what I want to show you here that what happened during this project. This project was a seedling to show the feasibility and the scalability of this whole concept. So animals are hosting the pathogens, mosquitoes are sampling the animals. The team developed a robotic mosquito trap, which was selected to the type of the mosquitoes. So it could zoom down for what is the mosquito, what needs to be collected. These traps were deployed in the environment or manually if the environment allowed, or using drones. The collected material was sequenced and then sent through a metagenomics pipeline. In the metagenomics pipeline, the different pathogens were sorted out, and not only known pathogens, but also those kind of sequences that were suspicious, that potentially dangerous for humans. They were also selected, and finally maps generated. So that was the whole project. We finished that project in 2016, and it was quite a success. Of course, as you know, in DARPA type of projects, what happens is that when a project finishes, then everyone declares victory, and then frequently the valley of death is coming. Now, in this case, valley of death didn't come because Microsoft was quite enthusiastic to promote further the whole project. In the next 4 years, until basically now, many things happened. Primarily, Microsoft Research developed, Ethan's group developed these different versions of robotic field biologists. They developed a number of general robotics modules to collect mosquitoes and other insects. They tuned the metagenomics pipeline for a very high performance pipeline, which is deployed now on the Azure cloud system. They conducted field tests in all sorts of cases in Africa, in the Caribbean, and in Texas. And we got today, now, obviously, this whole program has a huge significance in not only tracking an existing epidemics, but also predicting if something might emerge and provide an early indicator for that. Microsoft pushes further the whole project, and not only Microsoft, but we are also participating. Microsoft built for Ethan a fantastic lab for studying mosquitoes in an artificial environment. It's a level 2 lab. They run different kind of mosquito behavioral studies. There is a preparation now for a large-scale pilot in the Houston area. We are just engaged these days on organizing a new large projects. It will be a Microsoft plus a university teams project in which we are very heavily engaged in. And this project will be under the NSF Convergence Accelerator program, which is a very heavily DARPA-style program, which requires results to be delivered. And there are many, many other things that I cannot necessarily talk about it. And there are a bunch of requests for deploying these technologies in several places. So altogether, this is a fantastic example for an extremely cross-disciplinary project. It has huge impact, and there is a quite enthusiastic large team that is working on these, and we are pretty happy that one of our students lead this, and of course we are very deeply participating in every aspect of it. Thank you.
Thank you, Jonas. Thank you, everyone. I really appreciate your presentations. I know we have run a little bit over time, but I know about half of the Board of Visitors members are on this call, and I want to spend a few minutes to have the faculty members and the students answer any questions you may have.
Yeah, first of all, thank you very much. I'm very impressed with all the projects and how well you've engaged in such a short time. Dr. Webster, I'd like to ask, it sounds like with the FDA, that moved very quickly as far as agreement. And I was curious what that was like, or how did the FDA have the confidence so quickly in this case?
Well, so we submitted yesterday. We have not heard back from them. However, there are 2 other designs that squeeze ambu bags like ours does that have been put on the EUA, Emergency Use Authorization list, within the last 5 days. And those designs are like ours in one sense, not like ours in the sense that they can't be made in your garage. They require factories to make them. But they're like ours in the sense that they squeeze an ambu bag. So there are now 2 predicates that the FDA knows how to handle these things and will approve them. The reason we were able to go so fast with preparing the submission was the fact that I have a startup company that's doing surgical robotics, and we have regulatory people at the startup company who were willing to pitch in and work almost full time for like 2 weeks just on helping us prepare this documentation. And we were able to do animal studies at Vanderbilt so fast and get histology results and all of the testing results that you need. And so we'll see. I mean, it's in the FDA's hands now, and we'll see how it comes out.
It was really fantastic. The only thing that would make it better to use a Denso wiper motor. Thank you.
Indeed, we should have come to you. I don't know why I didn't think of that.
Other questions?
Of course, we should say that Britt gets some kudos as well with what he's been doing at his company. So thank you, Britt. That was fantastic.
Hey, it's Richard Schrader. Just today finished 60,000 that we finished production of 60,000 face shields, and those are all donated to hospitals. And TEMA has approached us now, and they're asking for 50,000. So we're gonna crank up the line again and get more out there.
Hey, John Perlin here. Just wanted to really offer kudos to Nick Adams for that work. As Chief Medical Officer at HCA, we see testing as the major bottleneck. And, this would be absolutely game-changing if borne out. And so I wish you every bit of luck with that project. It's really the most important immediate step in the control of COVID for a couple of reasons. First, the vaccine is at least a year off. And second, you have to be able to test for the virus, that is the antigen. The serology that tests for the antibodies, we're not really actually quite sure how to use that because we don't know that you actually get full immunity and you still may be shedding virus. So the only reasonable test at this moment to be able to isolate or quarantine individuals and to get back to business is the antigen test, which means testing for the virus. And the amplification time and preparation is really the long pole. So just really want to give a shout out for what's exceptional work among a series of presentations that are all exceptional work. So way to go.
Hey, it's Richard Schrader. Can you hear me?
Yes.
I got my Vanderbilt shirt on. I got my Vanderbilt cup. I'm all set to go here. Hey, Bob, it's good to see you. I'm glad you're working on some cool stuff, as always. But don't forget about us old guys with the prostrate, non-invasion surgery. I'm really watching for that one, okay?
Still going.
Okay, good.
All right, any other questions from the BOV members? Well, if not, I would like to thank everybody for being on this call. I'd like to thank the faculty members and their fantastic students and colleagues for the great work they do. Indeed, the speed at which they have been able to adapt in this very, very difficult world where basically we can't go into our labs in a regular way. I'd like to thank the Board of Visitors members for being here, and I'd like to thank everybody who put this call together. This call has been recorded and will be put on our website shortly, and will be advertised and made available to the entire School of Engineering community on Wednesday in my email conduit newsletter. With this, I wish you a good day, good end of the day, a great weekend, stay safe, and thank you very much for being great supporters of the school.